Thank you, Cal, and thank you, Emma and Bill, for inviting me to come back this year and present. And I don't know about you all, but anybody that can go through 65 slides in 10 minutes can be excused for missing an H. So I'm going to be talking to you today about AGTC. We're a publicly traded company, and here's our forward-looking statements. We are creating value for patients, number one, as well as for stakeholders. We are a gene-based company developing therapies in rare ophthalmology, CNS, and otology indications. Today I'm going to focus my presentation on the very exciting clinical results we were just able to present in both X-linked retinitis pigmentosa and in achromatopsia. But the value of the clinical data that we're being able to present are really based on the foundation of the company. We have a very strong technology platform. We were founded by 5 virologists, which means that we're very based on the data and the engineering of our product and specifying how that product is designed for each specific clinical indication. And I think that this is very, very important to how you develop a breadth of data, how you design your clinical trial, how you plan for success, not just in the clinic, but into the BLA and into commercialization. We do have that broad pipeline, as I talked about, and it's very important to understand that we've got the manufacturing situation under control. There's been several foot faults in gene therapy in the last year, year and a half, and we have a very strong manufacturing system that I'm going to also talk to you about today. So our core value is to keep the patient in mind. I was very pleased the panel before lunch talked a lot about how when you focus on the patient first, when you interact with the patients, you learn a lot that helps you develop the very best products that you can. And we've done this from the very beginning of our company, always involving the various patient groups in our discussions. And in fact, one of the clinical endpoints I'm going to talk to you about today in achromatopsia, we would never have developed the endpoint. We would never have known that this was an issue for the patients unless we'd been talking to them on a regular basis. And I think we all know that in the end, the products have to meet the need of what the patients want. And so this is something we always keep in mind at our company. This is our very comprehensive pipeline. As you can see, the top products I'm going to be talking to you about today are for X-linked retinitis pigmentosa and 2 different programs in achromatopsia. But we also will be entering the clinic quite soon with an optogenetic program. And we recently announced a program in dry AMD. So we're going to take all that we've learned about how to target the cells in the eye, how to design the right product, and move into a large market indication where there really are not any acceptable, very rigorously developed treatments for dry AMD. And we're very excited about moving into a larger market program. So now let's move on to our clinical programs. So the first is a potential treatment for X-linked retinitis pigmentosa. And this is an audience I probably don't need to describe the disease to, but there are about 20,000 patients in the US and EU that suffer from X-linked RP. And it's one of the most highly degenerative of the retinitis pigmentosa family, in that the children begin at a very young age to lose their night vision and their peripheral vision. By the time these patients are between 45 and 55, they're completely blind. It's really important to understand that this is a disease that people experience every day. Every day they can tell that their vision is getting worse and worse, and it starts in the periphery. And so as we were developing our clinical trial program, we wanted to be able to test this product both in the periphery and in the central area to see how complete of a product we could develop and bring forward. So I'm very pleased now to be able to present data from this clinical trial. This is the interim 6-month data. This is from 8 patients who were treated indeed in the periphery. And I think what you can see here is that the treated eye, which is the red eye in each of the dose cohorts, low, medium, and high, the treated eye is showing encouraging signs of having stable vision, while the untreated eye is showing a decline in vision as measured by visual fields. So this, again, is something very transformative for the patients to think about, that they could retain for long periods of time that peripheral vision. And we're very much looking forward to the later data that we're going to be collecting in early 2020 to see that this separation of the curve and this stabilization of peripheral vision is maintained. Now moving on to 9 patients that were treated centrally, and they were treated centrally at 2 different doses. Of those 9 patients, we had 4 that we considered to be responders, and what you're seeing here is the microperimetry, so the central perimetry data for the 4 responders. And what you can see is that in the treated eye in red, we are seeing an increase, so not stabilization, but an improvement in visual sensitivity in the responders, which were 50% of the patients treated, compared to the untreated eye. In addition, this is a very detailed example of that perimetry. This is one of the patients, actually in the mid-dose group, where you can see the treated eye on top. Over time, that visual heat map, not only is it getting larger, so there's a larger area of visual sensitivity, but the colors are getting brighter, which means that the amount of sensitivity is better and better compared to baseline, where it stays constant over time. The other 3 responders had similar patterns of either they got larger visual fields, brighter visual fields, or a combination of both. We also saw some very early signs. Remember, this is just 3-month data for these centrally treated patients, so very early in the course of treatment. And what we saw was some very encouraging signs and improvements also in visual acuity. And so the mean here is a +5, but we had some of the responders that were as high as 10 letters of improvement in visual acuity. So very encouraging, and we're looking forward to the 6-month data. So our next steps are to collect that 6-month data, and we'll be continuing our ongoing discussions with the FDA to lead up to our end of phase 2 meeting in 2020, and we hope to initiate our pivotal trial in 2020. Moving on to achromatopsia. Achromatopsia is an orphan indication. It's about 28,000 patients in the US and EU, but it's very different kind of clinical phenotype. So here, only the cones are affected. Cones provide you your color vision, your fine visual acuity, and your ability to see in light. All of these patients only see in black and white and shades of gray. They are all very uncomfortable in the light. They could not be standing here to save their soul with this bright light on them. And they also are all legally blind. So this is also a very severe loss of visual function. I'm showing here very early data on the one endpoint that we actually get directly that doesn't go through the reading center, and this is the endpoint I talked about earlier. It's a measure, a quantitative measure of light discomfort. This is the endpoint that the patients care about the most. It's the thing that affects their lives the most. And in the B3 trial, one of the 2 genetic forms, we saw 1 out of 3 patients in group 2 and 2 out of 3 patients in group 3 that saw improvements in light discomfort, and that was corroborated in the A3 trial, where again at that mid-dose, we saw 1 out of 3 patients seeing an improvement in the visual, in the light discomfort testing. So we're very encouraged that when we pull down all of the data from the 6-month time point, that it will continue in this very positive direction. So one of the things I did want to talk about, and I talked about at the beginning, is our manufacturing. So we have been working on manufacturing since the start of our development. We are now producing up to 4,000 ophthalmic doses in only 25 liters. It's fully scalable in single-use bioreactors. There's no animal-derived products. We've transferred this program to several different contract manufacturers, and this really can serve to really be able to scale up, control costs, and enable us to move into the dry AMD program that I talked about earlier. Finally, in the last few seconds, much of our team has been together for a period of time. We're very experienced in the technology of gene therapy, but recently we made 2 important hires. As we're thinking about the fact that we got this very encouraging data, we're moving into pivotal, and then hopefully for a BLA and commercialization, we brought on a new chief medical officer, Teresa He. She came to us from Aerie, who you heard about earlier today, and we're hopeful that her very, very strong experience in late-stage development, working with the regulators, and commercialization will help us move our products forward very efficiently. We also hired Brian Krex from Alexion, who has deep experience in commercialization, again, to prepare our team for the future and success of our products. So thank you all for your time and attention, and I look forward to discussing these programs with you at another time. Thank you.